So good afternoon, everybody. I would like to extend my sincere gratitude to GAABESU for giving this opportunity to present on this platform on very important topic of introduction of biotechnology for engineers, the role of AI/ML in biotechnology. To begin, I would like to introduce our academic unit and highlight the key research initiative undertaken to address the emerging healthcare problem. So the Center for Healthcare Science and Technology is introduced in the year of 2010 in erstwhile BESU. It is the complete interdisciplinary research and training program, and it basically encompasses the biomedical engineering, biotechnology, medicine, engineering, physics, chemistry, and different other fields for tackling the challenge of biomedical science and public health related problem. At present, we are offering course on the MTech and the PhD in the biomedical engineering. Our main thrust areas for research in the cancer biology, tissue engineering, neurotherapeutics, biosensor and bioelectronics, and the protein biology. So today I'll discuss some of the major research initiative that is right now undergoing in our laboratory, like the Translational and Cancer Diagnostic Laboratory. So the basic motto of this lab is to detect the cancer at very early stage. So we are mainly focusing to different cancers, oral and cervical cancer. And the main novelty of this research team is to detect the cancer in a non-invasive way, which is entirely a kind of very promising method if you compare with the traditional diagnostic system, which is the biopsy-based and very much invasive process. And that's why we cannot catch the cancer at very early stage, because a normal healthy person never been visit to the doctor for taking biopsy for the screening purpose. So the main motto is to screen the cancer in a non-invasive way. And another thing is the making a detection method without using any kind of biomarker, which is expensive and sometimes giving the subjective outcome. So one of the small research project that I'm sharing here, like this is for the oral cancer detection. So we all know that tobacco is a major cause of cancer, but sometimes it happens that the person who is taking tobacco for many years is not affecting with the oral cancer, and the person who is not even exposed to the tobacco is getting the cancer. So one project is taken like the taking up some cells from the oral cavity in a non-invasive way and see under the microscope if there is any subtle change at the cellular level. So these are some autofluorescence images, and which is actually showing in the last two row, that is the precancer and the oral cancer, which is already diagnostic in the laboratory, and we compared with the susceptible population who is exposed to the tobacco for a longer time period. And we also have taken different normal person who has no history of tobacco exposure. So this autofluorescence images is actually showing the metabolic activity of the cells. Further, when we confirm it with the established biomarker, we have seen that yes, some features we can identify from the autofluorescence images, and we can detect the cancer, or at least the onset of the cancer in the susceptible populations. So afterward, the different AI/ML based tool is integrated with it for differentiating the normal and the cancer cells. And if it is the cancer, what is the grade of the cancer? So some of the practical problem we are also facing while doing this thing, like when we are taking the cells from the mouth or the cervix for the cervical cancer, and we are making a smear on the slide, cells are actually overlapped. So when cells are overlapped, they are suppressing the feature of one cell to another cell. So we cannot get very good information from the cells out of it. So different AI/ML based tools are used for separating the cells without compromising their features. Then different spectral images, spectral data also obtained to get idea about what the chemical level or molecular level changes are going on. This is also a kind of without any biomarker or the level-free manner we are doing this thing. In another research work, prostate cancer detection was done and using the sonoelastography images. So sonoelastography, if we see, this is a kind of ultrasonography, but the main difference is that in sonoelastography, we are getting the stiffness property of the tissue. generally when the tissue becomes cancerous, it becomes stiffer. So how much the stiffness is, that can be detected, and based on that, some pseudo coloring or some AIML based tool is used for detect the cancer region and separate it from the benign or the non-affected person. So based on all our thing, but then another initiative is taken to make a very small microscope having this autofluorescence, having the capacity to measure the autofluorescence thing and integrate this AI/ML based detection algorithm for discriminating cancer and the normal cells. So our main aim is to provide this kind of setup to the primary healthcare sector or this kind of things where the or the resource limited areas, so the routine monitoring system can be done. So this thing project is funded by the DST as well as the ACRB, and a venture company PADUP just taken up our idea, and right now we are just going to register our first startup called Oncoscope, which is AI-powered multispectral microscope for prediction of epithelial cancer, mainly oral and cervical we are considering here. In another research venture, this is for the regenerative engineering and the mechanobiology lab. And here we use the stem cell for making any kind of graft. Graft means like when we have the deep injuries in the skin or even in the bone, then our normal healing mechanism cannot heal the entire wound area. So we need to provide some kind of artificial support, and which is we call the grafting. So grafting, the main problem is the donor availability is the main problem. So we are making some kind of artificial graft. The novelty of our graft is in this graft, we are trying to incorporate the blood vessel, because nowadays different type of grafts are available in the market, but they don't have any kind of blood vessels. So especially for the deep tissue injuries, if you don't provide the blood vessel or this kind of support, the healing process has become very slow. So for that, we isolate the stem cells from different biological waste, like the umbilical cord, which is generally thrown after the delivery, or the dental pulp from the deciduous tooth or the extracted tooth. And these stem cells are characterized by different means. Now, if you all know about the stem cell has the capacity to convert any kind of cells of our body, but it requires a different type of microenvironment. So this microenvironment is generally provided by means of different chemicals, different growth factors, nutrients, which are very expensive. So in a cost-effective manner, we are trying to modulate this microenvironment in the mechanobiological aspect, like by providing different type of stiffness, magnetic stiffness, or electrical stimuli to see how cells are actually behaving. So this is the image of the same stem cell isolated by in our laboratory, which are kept in two different stiffness conditions. So you see their morphology and shape is entirely different when they are growing in the different conditions. Now, similarly, when the cells is going inside the body, they are facing the interaction of the fluid. They are basically under the dynamic situation, but generally inside the laboratory, we maintain the static situation. So this is also one of our aim to see how cells are actually behaving under the dynamic condition. And in real-time imaging, we have seen that cellular morphology are actually entirely changing when it is facing any kind of dynamic interaction with the fluid instead of the static environment. So this using this method, using this concept, we applied 3D printing method for making the skin graft, and exosome, which is a kind of recent buzzword in the molecular biology. So we use make the bioink out of this exosome, which is also the novel one. And we also collaborate with the Aerospace and Applied Mechanics department to get the finite element analysis and the ANSYS images of the diagram that we are going to print. And this is the lab on chip on which we are just going to put the word scaffold to see how cells are behaving under the dynamic conditions. So these are different type of fabrication method, 3D printing method using different bioinks by providing biological molecules. We are real-time printing the layer of the grafts, and we see how cells are actually behaving when it is under the dynamic situation. So after 14 days of the culture, we have actually seen the formation of angiogenic sprout, that is the formation of the blood vessels, happen inside the skin graft, and which further we are just trying to apply for the animal studies. Our biosensor and bioinstrumentation lab is working in the field of development of the graphene-based sensor and other biosensor for the detection of the disease. And we are doing this work in collaboration of the electronics and telecommunication engineering. So one of the recent project right now we are working is the detection of the lung cancer from the biological fluid, again in the non-invasive way. And this is exosome based. So we are just putting the exosome on the graphene based biosensor and how this exosome is differ in normal and the cancer cell. This is the main aim of the study. So we have certain research facilities for the characterization and the fabrication of different scaffolds, and we have the state-of-the-art cell culture facilities for handling the cancer cell, normal cell, as well as the stem cell that we often isolate from different sources. We have another unit which is provided by the Ministry of Ayush, funded by the Ministry of Ayush for the fundamental research laboratory of the homeopathy, and this is a multi-crore project, and we got few state-of-the-art instrument under this grant, especially this confocal microscope with the TARP facility is present in our laboratory within the Eastern Zone. First time it is present in the laboratory. Few instruments are on the way, and when it will help us to get better information about the fundamental as well as the applied research. So some of the publication details for the last 5 years of our academic unit. So our research is funded by the different funding agencies by Government of India, and the net worth of the funding right now it's approximately 20 crores. Other than publication, few of the research are regularly cited by the different government agencies like the DST and DBT in their official website or the X-Handler to highlight how this research outcome is going on. So our faculty members and the students also acquire some recognitions in the national level. And these are the main recruiters who are working with us and recruiting our MTech student. Academically, our MTech and PhD students who are pursuing for the higher studies, they are now spread globally, and these are the few core institutes who are the takers of our alumni. So right now we have only 4 faculty members, but the positive thing is that the many departments right now working with us in collaboration for getting the interdisciplinary research and address different emerging healthcare problems. So this is a brief overview of our center and the research initiative, and thank you very much for this opportunity. Thank you.
Thank you, Dr. Barui. It's really fascinating to learn about the recent campus activities that are happening at IIEST. We really thank you for your presence here. Now, I would